Synthetic Biology and Phage Potential, narrated by Luke McLaughlin, biotech digital marketer, business developer and life science content creator. In the rapidly evolving landscape of biotechnology, synthetic biology emerges as a frontier with boundless potential, blending engineering with biology to reimagine solutions to some of our most persistent medical challenges. Among the most promising avenues within this field is the exploration of phage potential, viruses that can infect and replicate within bacterial cells. This exploration not only opens new pathways for treating antibiotic-resistant infections, but also heralds a new era of precision medicine. As researchers harness the capabilities of synthetic biology to engineer phage viruses, we stand on the brink of revolutionizing how we combat bacterial diseases, paving the way for targeted therapies that could transform patient outcomes worldwide. This article delves into the intersection of synthetic biology and phage therapy, exploring how these microscopic predators could potentially be the key to unlocking a future free of antibiotic resistance. Phage therapy is an exciting field that uses bacteriophages, viruses that infect and replicate within bacteria, to treat bacterial infections, particularly those resistant to antibiotics. Custom phage cocktails. Personalized medicine. Researchers are developing personalized phage cocktails tailored to individual patients' infections. This approach is crucial for treating complex antibiotic-resistant infections. Rapid matching. Technological advancements are enabling faster identification of phages against specific bacterial strains, significantly reducing preparation time for personalized treatments. Phage engineering. CRISPR-Cas systems. Phages are being engineered using CRISPR-Cas technology to enhance specificity and efficiency. This method allows the precise targeting of bacterial genes responsible for antibiotic resistance or virulence. Synthetic biology. Advances in synthetic biology facilitate the construction of phages with optimized therapeutic properties, such as improved host range and evasion of bacterial immune systems. Phage antibiotic synergy, PAS. Enhancing efficacy. Research has shown that combining phages with antibiotics can be more effective than using either treatment alone. Phages can disrupt biofilms and expose bacteria to antibiotics, reducing the overall bacterial load. Overcoming resistance, PAS has also been explored as a strategy to prevent the emergence of resistance, with phages selectively targeting resistant bacterial populations. Phage enzymes, endolysins. These are enzymes produced by phages that break down bacterial cell walls. Endolysins are being developed as topical treatments for infections like MRSA, and as potential replacements for traditional antibiotics. Engineered enzymes. Advances in protein engineering allow the modification of endolysins to enhance their bactericidal activity and stability. Biofilm targeting, disruption. Phages have a natural ability to penetrate biofilms, structures that protect bacterial colonies from antibiotics. This makes them particularly useful in treating chronic infections, such as those in cystic fibrosis or wound infections. Combination therapies. Studies are investigating the use of phages in combination with agents that disrupt biofilm integrity, enhancing overall treatment effectiveness. Regulatory and commercial development. Clinical trials. The number of clinical trials investigating phage therapy is increasing, focusing on safety, efficacy, and dosing strategies. Commercialization challenges. Despite promising results, the path to commercialization includes regulatory hurdles, given the complexity of phage therapy and the need for personalized treatment protocols. Global health initiatives. Non-traditional markets. In countries where regulatory frameworks are more flexible or antibiotic resistance is particularly acute, phage therapy is gaining traction as a feasible treatment option. Educational programs. Efforts are underway to educate clinicians and the public about the benefits and limitations of phage therapy to foster acceptance and proper usage. Synthetic biology and phage. Synthetic biology offers powerful tools for modifying and enhancing bacteriophages, making them more effective and versatile for therapeutic applications. Here's how synthetic biology is being employed with phages in the field of therapeutics. Phage engineering. Tailoring phages. Synthetic biology techniques allow for the custom design of phages with specific traits. For instance, genetic modifications can alter the host range of a phage to target a broader spectrum of bacterial strains or to focus very specifically on a pathogenic strain, minimizing impact on beneficial microbiota. Phage safety modifications. Phages can be genetically modified to include safety switches that ensure they only operate under specific conditions or can be deactivated externally. This reduces the potential for unintended consequences in therapeutic applications. CRISPR-Cas integration. Targeted bacterial killing. By incorporating CRISPR-Cas systems into phages, researchers can direct phages to cut specific DNA sequences within the bacterial genome. This allows for the precise targeting of genes that confer antibiotic resistance or virulence, thus neutralizing the bacteria's ability to cause infection. Minimizing off-target effects. Synthetic biology helps ensure that these CRISPR-equipped phages are highly specific to their target bacteria, reducing the likelihood of affecting non-target bacteria and preserving the host's microbiome. Improved delivery systems. Controlled release. Synthetic biology can be used to construct phage capsules or other delivery mechanisms that protect phages until they reach the specific site of infection, where they are then activated to release their therapeutic payload. Enhanced penetration. Modifications can also help phages better penetrate biofilms, which are often resistant to both antibiotics and traditional phage therapy. Combination therapies. Synthetic consortia. Designing consortia of phages that work synergistically to address complex bacterial infections can be achieved through synthetic biology. These consortia can combine multiple phages, each targeting different aspects of bacterial defense or different bacterial strains within an infection. Phage antibiotic combinations. Synthetic biology can enhance the effectiveness of phage antibiotic synergy, PAS, by engineering phages that release specific enzymes or chemicals that boost the efficacy of concurrently administered antibiotics. Phage-based biosensors. Detection and response. Engineered phages can also serve as biosensors that detect specific bacterial pathogens and respond by initiating a therapeutic action, such as releasing antibacterial agents or signaling to the host immune system. Therapeutic endolysins, engineered enzymes. Phages produce enzymes called endolysins that lyse bacterial cell walls. Synthetic biology can optimize these enzymes for better therapeutic properties, such as increased stability, enhanced lysing efficiency, or altered specificity to target different types of bacterial cell walls. Antibody-conjugated phages. There is significant interest in the development of antibody-conjugated phages for therapeutic applications. This innovative approach combines the targeting capabilities of antibodies with the antibacterial properties of bacteriophages. Here's an overview of the concept and current research directions. Concept of antibody-conjugated phages. Antibody-conjugated phages leverage the specificity of antibodies to target specific cells, tissues, or bacteria, fused with the ability of phages to deliver genetic material or other therapeutic agents. This strategy can enhance the precision and effectiveness of phage therapy, particularly in targeting bacterial infections that are difficult to treat due to their location or resistance to traditional antibiotics. Therapeutic applications, targeting specific bacterial strains. Antibodies can be designed to recognize unique markers on specific bacterial strains, directing the phage to these targeted bacteria. This is particularly useful for infections caused by antibiotic-resistant bacteria, or for targeting pathogenic bacteria without disturbing the rest of the microbiome. Enhancing phage delivery. The conjugation with antibodies can improve the efficiency of phage delivery to the infection site, especially in complex biological environments, such as tissues with poor accessibility, or where bacteria form protective biofilms. Research and development, oncolytic therapies. Some research has explored the use of antibody-conjugated phages in cancer therapy, where phages are used to deliver cancer-killing genes specifically to tumor cells. Although not targeting bacteria, this approach utilizes similar principles of targeted therapy. Dual functionality. Studies are investigating phages that not only carry antibodies for targeting, but also encapsulate drugs or therapeutic genes, providing a dual action against bacterial infections. Improved targeting and specificity. Research focuses on enhancing the specificity of the antibodies used, ensuring that the phages are directed precisely at pathogenic bacteria, reducing unintended impacts on beneficial bacteria and enhancing the therapeutic outcome. Challenges and considerations. Complexity in design and manufacturing. The design and production of antibody-conjugated phages involve complex biotechnological processes that require precise genetic engineering and quality control to ensure safety and efficacy. Regulatory and safety issues. The introduction of genetically modified phages into the body raises significant regulatory and safety concerns. Ensuring that these therapies are safe without unintended ecological or health impacts is crucial. Research integration. Combining phage and antibody therapies involves integrating knowledge from virology, immunology, and genetic engineering. Collaborative research efforts across these fields are essential to address the multifaceted challenges. Future directions. The field of antibody-conjugated phages is still in the early stages, with much of the work at the research or preclinical level. As both phage therapy and antibody therapy advance, the integration of these two fields promises innovative solutions to combat multi-drug-resistant bacterial infections and potentially other diseases like cancer. Overall, antibody-conjugated phages represent a promising frontier in therapeutic development, combining the advantages of phage therapy's efficacy and specificity with the targeting capabilities of antibodies. Aptamers conjugated bacteriophages. Yes, there is emerging interest in combining aptamers with bacteriophages to create novel therapeutic agents, particularly in the realm of targeted therapy. Aptamers are short, single-stranded DNA or RNA molecules that can fold into unique three-dimensional structures capable of binding with high affinity and specificity to specific targets, such as proteins, small molecules, and even cells. Concept of aptamer-conjugated phages. Aptamer-conjugated phages utilize the specificity of aptamers to guide phages to particular targets. This could be particularly advantageous in the field of targeted therapy, where such conjugates could deliver phages to specific bacterial populations or infected tissues, enhancing the selectivity and efficacy of phage therapy. Applications and research areas. Targeted bacterial infections. Aptamers can be designed to target specific bacterial species or strains, directing phages precisely to the pathogens responsible for an infection while sparing beneficial bacteria. This is highly useful in treating localized infections or infections caused by antibiotic-resistant bacteria. Drug delivery systems. By conjugating aptamers to phages, researchers can create systems that deliver therapeutic molecules directly to the site of infection or disease. This approach can be used to transport antibiotics, enzymes, or other therapeutic agents, potentially enhancing the therapeutic outcome by focusing the treatment directly on the affected area. Diagnostics. Beyond therapeutics, aptamer-conjugated phages can be used in diagnostic applications. They can help identify specific strains of bacteria rapidly and accurately, which is crucial for effective treatment planning. Current research and challenges. Specificity and binding. Research often focuses on identifying and optimizing aptamers that can bind with high specificity and affinity to target molecules. The challenge is to maintain this specificity and functionality when conjugated to a phage. Stability and delivery. Ensuring that the aptamer-phage conjugate remains stable and active in the human body is critical. Research includes developing methods to protect these conjugates from degradation by nucleases and other bodily enzymes. Regulatory hurdles. As with any advanced therapeutic approach, there are significant regulatory challenges. Ensuring the safety and efficacy of aptamer-conjugated phages through clinical trials is essential before these therapies can be widely adopted. Integration with existing therapies. Researchers are also exploring how aptamer-conjugated phages can be integrated with existing treatments, such as antibiotics or other antimicrobial agents, to enhance treatment effectiveness and possibly reduce the emergence of resistance. Future directions. The field of aptamer-conjugated phages is relatively nascent, with most studies still in the preclinical or early experimental stages. Future research will likely focus on enhancing the design and delivery mechanisms of these conjugates, expanding their applications not only in treating bacterial infections but also in targeted drug delivery and precision medicine. As technology advances, the combination of aptamers with phages could revolutionize how we target and treat various diseases, offering a promising avenue for overcoming some of the limitations of current therapeutic approaches. Antibody and aptamer conjugation to phages involves sophisticated biochemical methods that enable the precise attachment of these targeting molecules to bacteriophages. This conjugation enhances the specificity and therapeutic potential of phages. Here are the main methods and techniques used for these conjugations. Antibody conjugation to phages. Chemical cross-linking. Direct conjugation uses bifunctional chemical linkers like succinimidyl esters or malimids to covalently attach antibodies to amino groups on the phage capsid proteins. Indirect conjugation involves an intermediary molecule, such as streptavidin biotin complexes, to bridge the antibody and the phage. Genetic fusion. Phage display. Technology. Antibodies or their fragments, for example single-chain variable fragments and scFv, are genetically fused to phage coat proteins, typically at the tip of the phage, ensuring the antibody is presented on the phage surface. Affinity tag-based conjugation. His tag. Phage proteins are genetically modified to include histidine tags that can be used to attach antibodies that have been modified with nickel nitrilotriacetic acid, NiNTA. Biotin streptavidin. Phages or antibodies are biotinylated and subsequently conjugated via streptavidin as a linker. Aptamer conjugation to phages. Direct chemical conjugation, click chemistry, utilizes bioorthogonal reactions, like azide-alkene cycloaddition, to covalently attach azide functionalized aptamers to alkyne-modified phages, or vice versa. Thiol-melamide chemistry involves the introduction of thiol groups into aptamers and their subsequent reaction with malamide functionalized phages. Biological conjugation, genetic fusion. Aptamers are genetically engineered to be expressed as part of one of the phage coat proteins, ensuring that they are displayed on the phage's exterior. Linker fusion. A flexible peptide or nucleotide linker can be used to fuse the aptamer to phage proteins, promoting efficient exposure and functionality of the aptamer. Non-covalent conjugation. Electrostatic interactions. Utilizes the natural attraction between negatively charged nucleic acids, aptamers, and positively charged regions on phage capsids for attachment. Affinity tag. Similar to antibody conjugation, utilizing biotin, streptavidin, or similar systems to non-covalently attach aptamers to phages. Common considerations and techniques. Orientation and functionality. Both methods strive to maintain the orientation and functionality of the conjugated antibodies or aptamers. It is critical that these molecules are presented in a manner that does not hinder their binding capability. Purification and validation. Post-conjugation, it is necessary to purify the conjugated phages and validate their functionality and specificity. Techniques like affinity chromatography and ELISA for antibodies or binding assays for aptamers are commonly employed. Stability and sterility. Ensuring the stability and sterility of the conjugated phages is crucial, especially for therapeutic applications requiring rigorous quality control processes. These conjugation techniques are continually refined to improve the efficiency, stability, and therapeutic efficacy of phage-based treatments, particularly as the demand for targeted therapies grows in the medical field. In this exploration of cutting-edge biotechnological advancements, we've covered several innovative therapeutic strategies that harness the potential of bacteriophages in combination with other biological agents like antibodies and aptamers. These technologies represent a promising frontier in the battle against bacterial infections, particularly those resistant to conventional treatments. Phage therapy, leveraging the natural bacterial targeting properties of bacteriophages, has been significantly enhanced by synthetic biology. This approach not only improves the specificity and efficiency of phages, but also introduces engineered capabilities such as antibiotic synergy and targeted bacterial gene disruption. The integration of CRISPR-Cas systems within phages exemplifies the precision now achievable in targeting bacterial resistance genes, offering a hopeful strategy against multi-drug-resistant bacterial strains. The development of antibody-conjugated phages introduces a layer of specificity in targeting bacterial infections. By attaching antibodies to phages, researchers can direct these biological entities to specific pathogens, increasing the efficacy of the phages and minimizing off-target effects. This method holds particular promise in addressing localized infections and in settings where bacterial populations need to be precisely targeted without affecting beneficial microbiota. Similarly, aptamer-conjugated phages offer a novel approach by combining the targeting prowess of aptamers with the therapeutic potential of phages. Aptamers can be designed to bind with high affinity to specific molecular targets, guiding phages directly to the sites of infection or disease. This strategy is particularly advantageous for delivering therapeutic agents or enhancing the diagnostic capabilities of phages. In terms of technical implementation, various methods such as chemical cross-linking, genetic fusion, and affinity tag-based approaches are employed to attach antibodies and aptamers to phages. Each technique aims to preserve the functionality and orientation of these targeting molecules, ensuring that the conjugates are effective in their therapeutic roles. The convergence of these technologies highlights a transformative phase in medical biotechnology, offering innovative solutions to some of the most challenging problems in infection treatment and drug delivery. As research progresses, these approaches could significantly alter the landscape of therapeutic strategies, providing more precise, effective, and targeted treatments that could revolutionize the field of infectious disease and beyond. Stay curious, stay innovative. I'm Luke McLaughlin. If you would like to learn more about this topic, consider reading our full-length articles on biotechnologyreviews.com, or consider our podcast series on YouTube or Spotify at our Biotechnology Reviews channel. Our articles are also available on our LinkedIn newsletter, Biotechnology Reviews.